Welcome to the deep dive. Today we're doing something, well, a bit different.
Right.
We're setting aside the daily headlines, the newest virus variant, that specific symptom cluster, the latest trial for just one disease.
Mm-hmm.
Instead, we're going deeper. We're hunting for the really fundamental, the unchanging biological laws, the rules that govern health, disease, life and death, really.
That's a perfect setup because the source material you gave us treats science not just as like a list of facts, but almost as a philosophical quest.
Exactly.
And our mission today, for you listening, is to reach what these sources call abstraction de tercer grau, third-degree abstraction.
Okay, third-degree abstraction. That sounds pretty intense,
Yeah.
like something Plato or Aristotle would talk about.
Yep.
What level are we actually aiming for here? What does that mean?
Well, it's aiming for the highest level of understanding, really. It's about finding the knowledge that is necessary, the stuff that has to be true for the biology to work the way it does.
Necessary. Okay, so not just what happens to be true, but what must be true.
Precisely. We're trying to move past all the contingent factors, the variables, the things that change from person to person or from one virus to the next.
Right.
And instead, identify the fundamental, non-contingent biological laws.
Mm-hmm.
The rules that govern critical illness or, say, immune conflict, no matter the specifics.
Okay, give me an analogy.
Think about it like this.
Yeah.
Instead of just diagnosing one specific car crash, this model, this driver, we're trying to understand the universal laws of physics.
Like kinetic energy, structural failure, things that apply to any crash.
Exactly. The laws that govern the outcome, regardless of the particular details of the car or the road conditions, that's the level of abstraction we're aiming for.
So we're searching for the logos, is that the term they use? The sort of eternal blueprint for biological crisis.
That's the concept, yes.
Yeah.
The underlying invariant principle. But you're right, extracting that from the messy reality of clinical data, well, that's the
It
challenge.
just sounds incredibly difficult. How do the sources even approach that?
It takes a lot of methodological rigor. The sources, they draw on a blend of philosophical traditions, actually.
Oh, interesting. Like what?
They use what's called concrete philosophy, which means you start with the empirical data, the lab results, the patient charts, all the specific details.
Okay, standard science so far.
But, and this is the key, you look at that specific data with the explicit goal of finding the universal pattern within it. You're not just cataloging facts. You're searching for the law they illustrate.
Got it. Look at the particular to find the universal. What else?
And then they use speculative philosophy to build a hierarchy of understanding, to make sure that when we do find a pattern, we know if it's just like a correlation, something that happens alongside something else, or if it's a fundamental law, a necessary connection.
So sorting the signal from the noise, essentially, making sure we know what level of truth we've uncovered.
That's a great way to put it. The whole exercise is about building this verifiable bridge between the empirical world, our lived experience and observations, and the world of these necessary eternal biological forms, the logos.
Okay, this is fascinating. But for us to really grasp this, and for everyone listening, we absolutely need to unpack this core distinction. It seems like everything hinges on it. What is the difference between the essence of a disease and its accidents? If I miss this distinction, I feel like I miss the whole point of this deep dive.
You're absolutely right. That distinction is the key. It unlocks everything in these sources. So the accidental, these are the components, the factors that are contingent. They're mutable, variable.
Things that could have been different.
Exactly. They're crucial for treating you, the individual patient, right now. But they could have been otherwise, and the fundamental nature of the disease process wouldn't change.
Okay, so examples might be.
Like the specific virus that triggers it, or maybe exactly where the symptom shows up first. Things that vary.
Got it. Contingent, variable. So what's the essential?
The essential is the opposite. It's the core invariant mechanism, the biological principle that's universal and necessary. It's the thing that gives the disease process its identity.
The rule book itself.
That's a perfect analogy. The rule book itself. The accidental is the specific data point you observe in one game. The essential is the fundamental rule of the game that made that data point possible.
Right. Okay, I think I'm starting to get the philosophical framework. So let's dive in. We have to start, I suppose, in that messy, variable world of the accidental. That's where medicine operates most of the time, right? Dealing with things that depend on time, place, the individual.
Exactly. We have to map out the territory of the accidental before we can see the essential laws clearly. So yes, let's begin with the accidental. As we said, these are all the contingent, changeable factors we see everywhere in clinics and labs. Millions of data points.
Things that are critical for diagnosis, for managing a specific person's illness.
Absolutely critical for that. But crucially, they are not, in themselves, the fundamental law that governs the state of, say, critical illness or immune attack. They are expressions of the law under specific circumstances.
We need to understand these details, these variables, before we can abstract away from them, right? See the pattern through the noise. So you mentioned the source material breaks these accidental factors down into categories.
Yes, broadly into four useful categories. And the first one is maybe the most obvious and potentially the biggest distraction in public health discussions. It's the agent etiologico.
The etiological agent, so the specific trigger, the bug.
Exactly, the specific trigger for the crisis.
I mean, if someone is seriously ill, the doctor's first job is usually to figure out what caused it, isn't it? Is it SARS-CoV-2? Is it a particular strain of flu? Is it E. coli or staph or something else entirely?
Absolutely. Identifying the agent is step one in clinical practice. The sources use examples like pinpointing SARS-CoV-2 as the cause of COVID-19, or identifying a specific gram-negative bacterium in someone with sepsis. That's vital information.
But why is that accidental according to this framework? It feels pretty essential if that's what's making you sick.
Here's the philosophical twist from the sources. While the agent is the initial spark, the thing that initiates the whole event, it doesn't define the fundamental process of the critical crisis that follows.
Okay. So the specific virus, like SARS-CoV-2, is the accident. It's the match that lights the fire. But the wildfire itself, the body's catastrophic response, the cascade of failure, that's the essence.
You've got it. Think about it. If you swapped out that specific gram-negative bacterium for, say, massive physical trauma from an accident, or an overwhelming fungal infection, or even a different type of severe viral infection.
The end result, the way the body system starts shutting down, it often looks remarkably similar, doesn't it? Sepsis is sepsis in a way.
Precisely. The sequence of events, the systemic collapse, often follows a functionally identical path because the body's ultimate crisis response mechanism is conserved, it's standardized.
So the specific name of the microbe, the trigger, is just a label for the starting gun. It's an accidental detail.
It's the label for the starting pistol, yes. And the argument is, if we pour all our scientific energy into just chasing the label on that pistol, we risk missing the universal law that governs the race itself, the collapse.
That's a really powerful way to frame it. Okay, that distinction makes sense. What's the second category of accidental factors?
The second one is the manifestation local. Basically, where in the body the problem happens to show up first or most prominently, the specific anatomical location of the symptom or the lesion.
Location. Okay, so like if someone has pneumonia, the focus is the lungs. If they have meningitis, it's the brain lining.
Exactly. The sources give examples like localized pulmonary edema fluid in the lungs during a critical illness, which we often see. Or think about gout.
Right. Gout often presents with those painful crystals, the tophi in the big toe or maybe the hands.
Correct. Clinically, we focus intensely on that location, right? We treat the lung inflammation, we try to manage the pain and swelling in the joint. But the sources push us to ask, is the big toe the logos of gout? Is it the essential nature of the disease?
Intuitively, no. The essence must be the underlying metabolic issue, right? The problem with processing uric acid, leading to crystal formation somewhere, the big toe just seems like the unlucky spot where it happens to deposit.
Exactly. The big toe is the anatomical victim, not the core principle. The location is considered accidental because it's variable. It depends on secondary factors.
Like what?
Things like gravity, local temperature, maybe microvascular blood flow in that specific joint, or even previous minor injuries or wear and tear, making that spot more susceptible.
So the underlying mechanism of crystal formation and the inflammation it causes is essential, but where those crystals land is accidental.
Precisely. Same with the pulmonary edema example. The mechanism of fluid leaking out of blood vessels into the lung tissue and air sacs, that's part of the essential pathophysiology of vascular leakage in critical illness. But why the lungs fail at that specific moment, maybe more than the kidneys or the liver, is contingent on the initial insult, the patient's lung health, the pressures involved.
So the specific location becomes an accidental manifestation. It tells us where the essential process is happening, but not what that essential process is fundamentally.
You've nailed it. It points to the battlefield, not the rules of war.
Okay, trigger and location are accidental. Now, what about the differences between people? This seems like a huge source of variability. Variability individual. Why do two people exposed to the same virus have vastly different outcomes? One gets sniffles, the other ends up in the ICU.
This is where the sheer complexity of clinical medicine really lives. Individual variability covers a massive spectrum of factors that dramatically change how a crisis unfolds in a specific person.
What kind of factors are we talking about?
Well, the sources highlight things like differences in ACE2 receptor expression. We heard a lot about that with COVID-19.
Right, the receptor the virus uses to enter cells. Variations could affect how easily someone gets infected or how severely, especially, they noted, in adult males.
Yes, that was one observation related to severity patterns. Then there's the huge impact of pre-existing conditions or comorbidities.
Diabetes, heart disease, obesity, lung conditions. These clearly change the baseline health and how someone responds to a new stressor.
Absolutely. They profoundly alter the host's internal environment, making them more vulnerable or altering the inflammatory response. And then, of course, there's genetics.
You mentioned HLA type specifically. Can you unpack that a bit? What are HLAs and why do variations matter here?
Sure. HLA stands for human leukocyte antigen. Think of them as molecular billboards on the surface of your cells. They present little fragments of proteins, peptides, from inside the cell to your immune system, specifically to your T cells.
So they're showing the T cells what's going on inside. Anything foreign in here?
Essentially, yes. Now, there's huge genetic variation in HLA genes across the population. Your specific set of HLA molecules determines which peptides you can display effectively.
Okay.
So let's say a virus infects 2 people. Person A has an HLA type that binds a key viral peptide really strongly, presenting it very effectively to T cells. They might mount a massive rapid immune response.
Which could be good, clearing the virus quickly, or potentially bad if it's too strong, causing damage.
Exactly. Now, person B has a different HLA type. It might bind that same viral peptide very weakly, or maybe not at all. Their T cell response might be much slower or weaker, or target a different part of the virus.
Leading to a completely different disease course and outcome. Maybe the virus replicates more, or the inflammation pattern is different.
Precisely. So these HLA variations are critical in determining individual susceptibility and severity for many infectious and autoimmune diseases. They affect binding affinity, how strongly the peptide sticks to the HLA molecule for presentation.
Okay, that feels pretty fundamental to my personal risk, my outcome. If my genes make me likely to react severely to virus X, that seems essential to me. Why do the sources still classify this incredibly important individual variability as accidental?
It's a subtle but crucial philosophical point again. These factors, ACE2 levels, comorbidities, HLA types, are absolutely decisive for who gets sick and how severely. They dictate the degree of illness, the individual experience.
Yeah.
But they're still contingent variables. They describe the differences between people, the heterogeneity within the population. They explain why patient A's experience differs from patient B's.
Okay.
But the underlying fundamental biological laws, the rules of how inflammation works, how coagulation gets triggered, how T cells recognize peptides, those laws would still exist and operate even if, hypothetically, everyone had the same ACE2 levels and the same HLA type.
Ah, I see. The law itself isn't dependent on my specific genetics. My genetics are like the specific conditions under which that universal law plays out in my body.
That's the framing. The law is essential. The specific context or environment in which the law operates in an individual is accidental, from the perspective of defining the law itself.
Okay, that clarifies the divide. It's about defining the rule, not every possible application of the rule.
Got it.
Got it. So that brings us to the fourth accidental factor. You called it an epistemological trap. Correlation. Temporal. Simple timing.
Yes, temporal correlation. This is less about biology itself and more about how we interpret events. It's a critical point for scientific reasoning.
What is it? Simply.
It's just the observation that event A happened, and then, sometime later, event B happened. That's it. Just an association in time.
That observation is necessary to even start thinking about whether A caused B, right?
Absolutely necessary to start an investigation. But, and this is the crucial part stressed by the sources, it is completely insufficient on its own to conclude causality.
This feels like the most contentious issue in public health discussions right now, especially around things like vaccines or new medications.
Definitely.
If someone takes a new drug and a week later develops a rare condition, the human brain almost instinctively leaps to the drug caused it, even without any biological proof.
It's a very natural cognitive bias, linking events that occur close together in time. But the sources are adamant. Without proof of a plausible, demonstrable biological mechanism connecting event A to event B, the temporal association is merely accidental. It's a coincidence until proven otherwise.
So it tells us when and what happened, but provides zero information on how or why.
Exactly. It's the weakest form of evidence when you're hunting for an invariant necessary law, the logos. It might prompt a hypothesis, but doesn't confirm it.
So to move from just noticing that B followed A, correlation accidental, to proving that A caused B, causality essential,
Yeah.
we need something more. We need the mechanism.
Precisely. And that need for mechanism leads us directly out of the world of the accidental and into the world of the essential laws.
Okay. Wow. We've really mapped out the terrain of the accidental now. The specific triggers, bugs, trauma, the specific location of damage, the vast individual differences in response, and the trap of just looking at timing. It's messy, complex, contingent.
It is. It's the world of clinical practice.
But now,
Yep.
now for the exciting part.
Yeah.
We climb up, as you said, Aristotle's ascending path. We leave the messy details behind, seeking the pure universal laws that dictate the outcomes, despite all that variability. Time to talk about the essential.
It's exactly. Welcome to the world of the essential. Now we're aiming for that abstraction de tercero grau. We're looking for the principles that are universal, invariable, and necessary. The things that define the phenomena's identity. It's logos.
These are the fundamental rules of the game, conserved across different players and situations. So when we reach this third degree of understanding, what's the first essential law we find? What's the core, standardized way the body reacts when facing a truly critical threat, regardless of what accidentally triggered it?
The first essential principle is the risposta di crisi universal, the universal crisis response. It's the conserved, almost pre-programmed pattern of biological reaction that defines what it means to be critically ill.
Okay, so what does that standardized pattern actually look like? What are the components?
At the highest level, we're talking about the final common pathways that lead to systemic failure. Specifically, the sources emphasize two interconnected syndromes: systemic inflammatory response syndrome, SRS, and disseminated intravascular coagulation, DIC.
SIRS and DIC. These terms come up constantly in critical care.
They do. And the insight here is that this pairing represents a massive simplification of critical illness, but it's a profoundly true simplification. Whether the initial insult, the accidental trigger, was a horrific burn, a bloodstream infection, severe pancreatitis, or a virus like SARS-CoV-2, once the body's systems are pushed past a certain threshold.
The way they collapse starts to look the same.
Yes. The trajectory towards multi-organ failure very often involves these conserved syndromes. The specific agent is accidental, the host's runaway, hyperreactive SIRS response, often progressing to DIC, is essential. It's the body's stereotyped way of failing.
That really drives home the point that the essence of the crisis lies within the host's response, not just the identity of the invader or injury. Can you briefly define SIRS and DIC in practical terms, just to show how this collapse is recognizably similar across different causes?
Sure. SIRS, systemic inflammatory response syndrome, is basically the body's inflammation going into overdrive, systemically. Clinically, it's often defined by having two or more signs like abnormal body temperature, high or low, a very high heart rate, rapid breathing, and an abnormal white blood cell count. It's the body sounding all alarms everywhere, non-specifically.
A massive uncontrolled inflammatory reaction, and DIC, disseminated intravascular coagulation.
DIC is the catastrophic breakdown of the clotting system that often follows severe SIRS. It's paradoxical and deadly. The body starts forming thousands of tiny, inappropriate blood clots throughout the small blood vessels.
Okay, clotting everywhere. That sounds bad. It blocks blood flow.
Yes, it blocks microcirculation, damaging organs. But here's the paradox. Because you're using up all your clotting factors and platelets to make these tiny clots everywhere, you can simultaneously start bleeding uncontrollably from other sites because there's nothing left to form a normal clot where you do need one.
Wow, so widespread clotting and bleeding at the same time. It's a complete system failure.
It is. It's like a self-destruct sequence. And the universality of this SIRS to DEC pathway, this conserved script for disaster, points directly to the next essential law, the mechanismo pathogenetico chave.
The key pathogenetic mechanism.
Yeah.
So what's the underlying engine driving this standardized SIRS-DIC crisis? What's the core connection between the systems?
This is really the central destructive principle, the physics of the collapse. The sources define it very clearly as the inflammation thrombosis axis, or, captured in a single powerful term, inflammothrombosis.
Inflammothrombosis. Okay, this sounds incredibly important. We need to spend some time on this because if I understand you right, this concept potentially unifies thousands of different disease outcomes down to one core destructive process. Can you describe this axis, this loop?
Absolutely. The logos here, the essential truth, is the complete and utter interdependence between the immune system inflammation and the coagulation system, thrombosis or clotting. Historically, medicine often studied these as separate fields,
Right, immunology and hematology.
but they are intrinsically fundamentally linked at the molecular level. In a crisis state, like severe infection or trauma, the inflammatory molecules cytokines released during SIRS don't just fight infection, they directly activate the endothelial cells.
Those are the cells lining the inside of all our blood vessels.
Exactly. And critically, inflammatory signals flip these endothelial cells from their normal state, which is anticoagulant, preventing clots, to a highly procoagulant state.
So the inflammation itself is telling the blood vessels, start making clots. It's turning the entire vascular system into a potential clotting factory.
That's a perfect way to put it, yes. This widespread activation leads to the formation of microthrombotic clots, which is the hallmark of DIC. And as you said, these clots block blood flow, starving tissues and organs of oxygen.
Okay, that's bad enough. But you called it a loop, a vicious loop.
Yes, here's the truly destructive part. The act of forming those clots and the damage caused by the lack of blood flow, ischemia, itself triggers the release of even more inflammatory molecules. Damaged cells spill their contents, activated platelets release pro-inflammatory signals.
Which then fuels even more inflammation.
Precisely. And that new wave of inflammation further activates the endothelium, driving more coagulation.
Oh, wow. It's a positive feedback loop from hell. Inflammation causes clots. Clots cause more inflammation, which causes more clots.
Exactly. It's an accelerating, self-sustaining cycle of destruction that spirals towards multi-organ failure. That's inflammothrombosis.
So the inflammothrombosis mechanism is the essential engine of critical illness. If you understand this axis, you understand the universal way the body fails catastrophically, no matter what accidental event kicked it off.
That's the argument from the sources, yes. It provides the fundamental why behind the observed SIRStic pattern.
And this essential engine, this destructive loop, it must be driven by specific molecules and cells, right? We need to go one level deeper. What's the third essential law? The mechanismocellular molecular. What's the physical bridge between inflammation and clotting?
Right. We need the molecular hardware. And the sources point compellingly to one key player that perfectly embodies this bridge, neutrophil extracellular traps, or NETs.
NETs. Okay, tell me about neutrophils first.
Neutrophils are the most common type of white blood cell, our frontline infantry against bacteria and fungi. Usually they engulf pathogens or release toxins. But when faced with an overwhelming threat or massive systemic inflammation, they can do something drastic called netosis.
Netosis, what's that?
They essentially commit cellular suicide, but in a very specific way. They deliberately rupture, spewing out their nuclear contents, their DNA, into the surrounding space.
They throw out their DNA. Why?
That DNA, along with various proteins and highly toxic enzymes like histones and elastase, normally kept inside the nucleus, forms a sticky, web-like structure. That's the net.
Physical net to trap pathogens.
Yeah, exactly. It's designed as a localized defense mechanism to physically ensnare and kill microbes, preventing them from spreading. It's actually quite ingenious in a local infection, but in a systemic crisis, like severe sepsis or major trauma, when neutrophils are massively activated everywhere and start throwing out these nets uncontrollably throughout the bloodstream, they become incredibly destructive.
Okay, how do these sticky DNA webs bridge the inflammation and coagulation parts of inflammothrombosis?
The sources are very clear on this. Nets are profoundly procoagulant. They drive clotting in multiple ways.
Oh.
First, the DNA scaffold itself, this long, sticky mesh, acts as a physical surface that promotes thrombus formation. Platelets stick to it, clotting factors get activated on it. Second, those toxic proteins studded onto the nets, especially histones, they directly damage the endothelial cells lining the blood vessels. And as we just discussed, damaged or activated endothelium immediately triggers the coagulation cascade. Nets provide both the physical scaffold and the biochemical trigger for clotting.
Wow. So nets aren't just a byproduct of inflammation. They are an active driver of the thrombosis part of inflammothrombosis. They are the physical link.
That's the understanding. They are a perfect molecular manifestation of that destructive axis.
So let's connect this back. The accident might be a lung infection or maybe widespread tissue injury. But the essential law, the thing the philosopher or the deep biologist cares about, is the conserved mechanism of netosis itself. Because that mechanism is inherently prothrombotic and destructive, regardless of what initially triggered the neutrophils.
That is the impeccable logic, yes. The mechanism is conserved. And the proof that it's essential, not just tied to one disease, its universality.
Meaning we see nets causing problems in many different conditions.
Exactly. Net-driven clotting isn't just seen in severe infections like COVID-19 or sepsis. It's also observed driving thrombosis in non-infectious inflammatory conditions, like certain autoimmune diseases, lupus, vasculitis, deep vein thrombosis, DVTs, even potentially in atherosclerosis.
So the molecular machinery of netosis and its procoagulant effect is a fundamental biological process, reused or dysregulated in various disease states. That makes it essential.
Correct. It's conserved across disease categories.
Fascinating. Okay, we've established the essential laws governing systemic inflammatory failure. SIRSDIC, driven by the inflammothrombosis axis, with nets playing a key bridging role. Now let's pivot. Let's talk about the adaptive immune system, T cells, B cells, and the potential for friendly fire, for autoimmunity. What's the fourth essential law here? The Principio de Reconocimiento do TCR?
Right. This principle digs into the fundamental rules governing how our adaptive immune system, specifically T cells, recognize threats, and crucially how this recognition system inherently carries the risk of mistaking self for non-self. It's about the physics and mathematics of T-cell receptor TCR interaction.
The T-cell receptor, the molecule on the T-cell surface that sees the peptide presented by the HLA molecule we talked about earlier.
Precisely. This gets into some structural detail, but it's absolutely vital for understanding the logos, the essential nature of adaptive immunity and autoimmunity.
Okay, let's break it down. What's the universal non-contingent law here?
The core law, as laid out in the source material, is the principle of TCR degeneracy, or sometimes called TCR cross-reactivity or promiscuity.
Degeneracy. That sounds negative, like it's broken.
It sounds negative, but it's actually essential for function. It means that a single T-cell receptor is not exquisitely specific to just one single peptide sequence it first encountered. It's designed to be flexible, to recognize a range of similar peptides.
Why would that be necessary? Why not perfect specificity?
Think about viruses mutating rapidly. If your T-cells were only programmed to recognize the exact original sequence, a tiny mutation could let the virus escape immune detection completely. Degeneracy allows the T-cell to still recognize slightly mutated versions.
Ah, so it's a feature, not a bug. Built-in flexibility for broader protection. But that flexibility must come with a risk.
Exactly. And that risk is the crux of this essential law. The specific rule derived from structural studies is stunningly precise. For a T-cell to cross-react, meaning it gets activated by a new peptide that it wasn't originally trained on.
Yeah.
That new peptide, when presented by the same type of HLA molecule, only needs to share 5 specific amino acids in the right positions with the original activating peptide to trigger a response from that T-cell.
Wait, only 5 shared amino acids out of how many in total that the TCR interacts with?
Typically, the TCR might interact with, say, 8 or 9 amino acids of the peptide sticking out of the HLA groove. But the critical recognition, the triggering event, seems to boil down to matching just 5 key contact residues.
Just 5 points of contact need to match. That seems incredibly permissive. Flexible, yes, but also wide open for mistakes.
That's exactly it. This structural rule dictates the inherent permissiveness, or degeneracy, of our T-cell recognition. It makes the system adaptable enough to handle evolving pathogens, but it simultaneously creates an unavoidable, built-in risk of recognizing a self-peptide that happens to share those 5 key amino acids with a foreign one.
Molecular mimicry.
Molecular mimicry is the inevitable consequence of TCR degeneracy.
Okay, let's visualize this because you mentioned anchor residues and contact residues before. We need to nail this distinction.
Right, let's use that lock and key analogy again. The HLA molecule groove is the lock. The peptide is the key. To fit into the lock, the key, peptide, needs certain teeth, amino acids, to fit deep into the lock's tumblers. These are the anchor residues.
They hold the peptide securely in the HLA groove. Are they visible to the T-cell?
Mostly no. They're buried deep inside the groove. They are necessary for the peptide to be presented, but the T-cell receptor floating by outside doesn't really see them. So from the perspective of T-cell recognition, the anchor residues are accidental.
Okay, so what does the T-cell receptor see?
It scans the part of the peptide key that sticks out of the lock. The exposed surface. These are the contact residues.
And the essential law is that the T-cell only really cares about 5 specific points on that exposed surface.
That's the rule derived from the structural biology. If a different peptide, maybe from one of your own body's proteins, gets loaded onto the same HLA lock, and it happens to have the same 5 amino acids sticking out in those critical contact positions.
The T-cell will react. It thinks it's seeing the original foreign threat.
Yes. Even if the rest of the peptide, including the anchor residues holding it in the groove, is completely different. The T cell gets triggered based on that 5 amino acid match at the contact points.
So the specific protein sequence of the original pathogen, that's accidental. But the structural rule dictating that only 5 contact residues matter for triggering cross-reactivity, that's the essential law, the logos of T-cell recognition.
You've got it. That explains why autoimmunity isn't just some random, bizarre malfunction. It's a predictable structural inevitability baked into the system's design. The risk is inherent because the flexibility is necessary.
So the logos of potential autoreactivity is TCR degeneracy, the rule of 5 amino acids. It's a fundamental structural feature independent of which specific pathogen or self-antigen happens to be involved in any particular case. The risk is always there because the rule is always active.
Precisely. It's an essential property of the adaptive immune system.
Wow. Okay. We have really ascended now. We've moved from the messy ground level of specific triggers, locations, and individual responses up to these 4 fundamental essential laws. The universal crisis response, characteristically, driven by the engine of inflammothrombosis, powered molecularly by nets, and the inherent risk of immune cross-reactivity governed by TCR degeneracy. What's next? How do we solidify these insights?
Now we formalize them. We move from identifying these essential laws to stating them as apodictic postulates, necessary truths derived from the evidence. Right. This final section is about establishing the apodictic postulates. These are statements that the sources argue are necessarily true, derived directly from the concrete evidence of the essential mechanisms we just discussed in Part 2. This is where we demonstrate the logica impeccable, the impeccable logic that cements the findings at that 3rd degree of abstraction.
Apodictic, meaning like demonstrably true, beyond doubt within this framework.
Exactly. Not just probable, but necessary conclusions based on the established biological laws.
Okay, let's take them one by one. Postulate I, the invariance of the systemic inflammatory crisis. This sounds like it formalizes our discussion about surristic and inflammothrombosis being the common endpoint.
Precisely. It starts with the observation, which is incredibly consistent across medicine. Wildly different initial triggers, viruses, bacteria, massive injuries, autoimmune flares, you name it all, ultimately converge onto a remarkably similar, reproducible syndrome of systemic inflammation and organ failure.
They start differently, but they end the same way, pathologically speaking. Like, severe sepsis looks a lot like severe COVID-19 ARDS in the end stages, or like the response to massive trauma.
Yes, the endpoint failure state is conserved. Now, the impeccable logic follows directly from this observation. If many different distinct causes, the accidental triggers, reliably lead to the same unified effect, the conserved syndrome,
then the core essence of that effect must reside in the shared response mechanism, not in the unique nature of each individual trigger.
Exactly. The logic forces us to conclude that the defining characteristic of the crisis is the host's reaction pattern. So the law derived from this is that the essential conserved biological principle is the uncontrolled activation of that inflammation-coagulation axis, the process of inflammothrombosis.
And we need to be specific about the mediators involved to really ground this law, right? It's not just vague inflammation.
Absolutely. To make this rigorous, the sources emphasize that this conserved destructive cascade involves specific, measurable molecular pathways. We see the cytokine storm, characterized by massive release of key pro-inflammatory mediators.
Which ones are consistently implicated across different triggers?
Molecules like interleukin-6, IL-6, tumor necrosis factor alpha, TNF-alpha, are classic players. The courses also specifically highlight the IL-17 and IL-23 axes as being critically involved in driving this kind of damaging systemic inflammation in various crisis states.
So the fact that these specific cytokine pathways are predictably and destructively activated, whether the initial insult was bacterial LPS or viral spike protein or damaged tissue, provides concrete evidence for a universal conserved response pathway.
Yes, the signaling pathways that switch on these specific cytokines represent the necessary conserved element that defines the essential response.
Okay, so the final statement, the logos of crisis, the 3rd degree abstraction here is, the inflammothrombosis mechanism, driven by these conserved molecular pathways, SR, specific cytokine axes like IL-6, IL-17-23, is the necessary causal principle that governs the transition from a specific accidental starting disease state to the universal essential outcome of organ failure.
That's the postulate. And recognizing this essential mechanism is crucial because it suggests that treatments targeting this core inflammothrombosis pathway might be universally beneficial in critical illness, regardless of the initial trigger.
Targeting the essential, not just the accidental. Makes sense. Okay, let's move to the second postulate. Postulate 2. The permissivity of immunological recognition. This sounds like it formalizes the rule of 5 for TCRs.
It does. Again, it starts with an observation. T-cell cross-reactivity is not a rare anomaly. It's a common, experimentally demonstrable phenomenon. Classic research models like experimental autoimmune encephalomyelitis, EAE.
That's a model used to study multiple sclerosis, right?
Yes. In EAE, scientists can reliably induce autoimmune attacks on the nervous system by injecting animals with specific peptides that are similar, but not identical, to self-proteins in myelin. This clearly shows that total molecular identity isn't needed for a T-cell to get activated and cause damage. Cross-reactivity is inherent.
So the observation is that the system is inherently promiscuous or permissive. What's the impeccable logic flowing from that?
The logic is rooted in the structural biology we discussed. If the T-cell system routinely reacts to peptides that only partially match its original target, then the defining feature of recognition cannot be the entire peptide sequence. It must be something more limited, more specific to the interaction interface.
It has to be based on those exposed contact residues.
Precisely. The logic dictates that T-cell specificity is primarily determined by those few amino acids sticking out of the HLA-MHE groove, the contact residues. This makes the recognition process inherently degenerate or permissive.
And the specific quantitative rule that emerges from this
Is that the ability of a given TCR to react to different peptides, provided they bind the same HLA-MHC and share just those 5 critical amino acids in the contact positions, isn't just an observation anymore. It's established as a structural and mathematical law governing the adaptive immune response. It's a consequence of the molecular geometry of the TCR peptide MHC interaction.
It's a physical constraint, a rule of the system. And let's reinforce that crucial distinction again. The anchor residues, buried deep, holding the peptide in the groove.
They are accidental to the T-cell's recognition event. They're necessary for presentation, but invisible to the TCR.
Well, the 5 exposed contact points carry the essential weight for determining if that T-cell gets triggered or not.
Correct. Therefore, the logos of autoimmunity, the third-degree abstraction derived here, is TCR degeneracy, embodied by the rule of 5 amino acids. This is the universal principle. It establishes the intrinsic, unavoidable risk of molecular mimicry and autoimmunity as a fundamental property of the system.
A risk that exists regardless of the specific accidental trigger, whether it's a virus, a vaccine component, a bacterium, or something else entirely.
Exactly. The trigger is accidental. The underlying structural rule creating the potential for cross-reactivity is essential and always present.
This completely reframes autoimmunity. It's not a mistake in the usual sense, but an inherent consequence of the system needing to be flexible. It's the price of adaptability.
That's a very insightful way to put it. It suggests we should anticipate and manage cross-reactivity as an expected feature rather than being surprised by it. It has huge implications for vaccine design, understanding autoimmune diseases, and even cancer immunotherapy.
Absolutely. Okay, this brings us to the third and final postulate. This one sounds very practical, focused on scientific proof itself. Postulate the third. The priority causal of mechanism in vaccinology, tackling causality versus correlation head-on.
Yes, this postulate addresses the critical epistemological challenge of establishing true cause and effect, particularly in complex areas like evaluating vaccine or drug safety. The starting observation is a practical necessity in the field. Regulatory agencies and scientists constantly need to distinguish between adverse events, AAEs, that just happen after an intervention, temporal correlation, and those that are actually caused by the intervention.
And doing that rigorously requires putting less weight on simple timing.
And much more weight on demonstrating a plausible biological mechanism. The observation is that temporal association alone is insufficient proof.
Okay, what's the impeccable logic here?
The logic confronts the inherent weakness of anecdotal evidence or simple observation. If one person, or even a small group, experiences an unusual health event weeks or months after receiving, say, a vaccine, that raw empirical data, the case report or case series, is inherently contingent.
Meaning it could be due to chance or some other underlying factor. It doesn't automatically prove the vaccine caused it.
Exactly. That observation alone is insufficient to establish a necessary, irrefutable causal link, the logos of causality. Too many other variables are uncontrolled.
So how do we bridge that gap? How do we move from the accidental observation of B happened after A to the essential truth A caused B?
The sources are absolutely categorical on this point. That transition from mere probability or association, accidental, to demonstrated causality,
Yeah.
essential, can only happen through establishing biological plausibility.
Meaning,
Meaning, we need a demonstrable step-by-step physiopathological mechanism shown through control experiments in vivo or in vitro studies that explains exactly how the intervention A could plausibly lead to the observed outcome B.
so back to the example. If the adverse event is, say, myokinitis after an mRNA vaccine, simply noting that some cases occur after vaccination isn't enough. We need studies showing how the vaccine components might trigger inflammation in heart muscle cells.
Mm.
We need the molecular steps.
Precisely. You need to demonstrate the pathway. Maybe it involves specific immune cell activation or cytokine profiles or direct effects of vaccine components on heart tissue in experimental models. Without that mechanistic explanation, the link remains correlational, not necessarily causal.
And the sources mention specific methodologies used to help make this distinction.
Yes, they note that rigorous methodologies, like applying established causality algorithms, such as the Bradford-Hill criteria, or WHO algorithms for AEI assessment, are crucial tools. These algorithms don't discover the mechanism, but they provide a logical structure to evaluate all the available evidence, including temporal association, strength of association, consistency, specificity, and critically biological plausibility to help reason towards a judgment about causality.
They impose logical rigor on the interpretation of messy data.
Exactly. They are tools reason uses to find the essential mechanism hidden within the multitude of accidental observations.
Okay, so this leads to the final essential law, an epistemological one about scientific knowledge itself. The logos of causality is
The demonstration of the physiopathological mechanism, biological plausibility. That demonstration is the essential epistemological principle required to validate a true cause and effect relationship between an intervention, like a vaccine or drug, and a clinical outcome. It allows us to transcend mere temporal observation and establish necessary connection.
It's what separates scientific proof from coincidence or conjecture. It gives science its reliable foundation.
It insists that we're not just reporting what happened, but demonstrating the necessary molecular or cellular reason why it happened. That's the standard for establishing essential truth. Hashtag tag outro.
Wow. This has been, well, quite a journey, a really layered deep dive. We started grappling with some pretty abstract philosophy, distinguishing the accidental from the essential.
The contingent from the necessary.
And we ended up formalizing 3 incredibly powerful, fundamental, non-contingent biological laws that seem to govern huge swaths of human disease and immunology.
It's worth bringing those 3 big aha moments together again, just to crystallize them for you listening. These are the core takeaways if you want to operate at that 3rd degree of abstraction.
Okay, let's summarize. Takeaway number 1.
First, the universal law of systemic failure. Regardless of the accidental trigger that starts the fire, severe critical illness converges onto the inflammothrombosis axis. That destructive loop of inflammation driving clotting and clotting driving inflammation is the necessary causal principle behind multi-organ collapse. Understanding that mechanism is key.
Got it. Law number 2. Immune recognition.
Second, the law governing immune structure and risk. The potential for autoimmunity, for molecular mimicry, isn't a random error. It's hardwired into our adaptive immune system by the essential structural requirement of TCR degeneracy, specifically that rule of 5 amino acids. Cross-reactivity is a necessary consequence of the system's need for flexibility.
So inherent risk is baked in. And the 3rd takeaway, the law of proof.
And third, the epistemological law of scientific causality. In a world awash with correlations and anecdotes, especially concerning interventions like vaccines or drugs, the only essential principle that truly validates a cause and effect relationship is biological plausibility, demonstrated through a proven, step-by-step physiopathological mechanism. Mechanism trumps mere timing.
Those 3 laws, inflammothrombosis, TCR degeneracy, and the primacy mechanism, they provide a really solid, robust foundation for medical thinking, don't they?
They absolutely do. The specific symptoms you see today, the particular virus variant in the news, the exact timing of an event, those are the accidental facts, the day-to-day clinical reality we have to manage.
Yeah.
But our real understanding, our ability to predict, to intervene effectively across different diseases, our scientific mastery that rests entirely on recognizing, understanding, and eventually maybe even manipulating these underlying essential unchanging laws.
The sources really argue that while we're busy chasing the latest accidental manifestation, a new symptom cluster, a specific mutated protein, the body is still operating according to these ancient essential rules.
That's right. It's still collapsing via SIRS and DIC, driven by inflammothrombosis. It's still making clots fueled by nets. The immune system is still deciding friend from foe based on that 5-amino acid rule. The essence of biological danger, the fundamental mechanisms of failure and conflict, remain constant beneath the surface waves of specific diseases.
Which leaves us, and you listening, with a final provocative thought to chew on. Consider the immense resources, time, money, brain power, currently dedicated to chasing the accidental, cataloging every minor symptom variation, sequencing every single new viral mutation, tracking every temporal association. It's a staggering amount of effort. The question raised by this framework is, to what extent does this relentless, necessary focus on the variable, the contingent, the accidental, potentially distract us from achieving true mastery over the underlying essential laws, the laws of inflammothrombosis and TCR degeneracy? Aren't those the truly universal targets we should be aiming for if we want breakthroughs with global, long-term therapeutic impact across many diseases?
Understand the logos, the essential blueprint of how things fail or go wrong. That's what gives us the power to design truly fundamental interventions, to move beyond just reacting to the latest accidental crisis and towards manipulating the core mechanisms themselves.
Food for thought, indeed. Understanding the architectural blueprint of failure. I like that.
That's a powerful perspective.
Thank you for guiding us through this incredibly insightful, philosophically rich, deep dive into biomedical science.
My pleasure. It's crucial thinking.
And for everyone listening, go forth and seek the universal within the particular. Thanks for joining us on the deep dive.